All right, good morning. No, it's not morning. Good afternoon. Hope you all had a good weekend. I wanted to start this time just with a little bit of fun here. I wanted to show you, so we left off last time talking about pasteurization, and I think that I said something about chunky milk and how you shouldn't be afraid to drink milk that was chunky. So I wanted to show you, this is the best Super Bowl commercial of all time here. You won't be able to hear it very well.
$20 a month. That's not right.
It's right, all right. Not chunky style milk, that's not right.
I choose chunky style milk because it has the wholesome chunks growing kids need, unlike smooth style
milk.
Hey guys, save some
Take
chunks for me.
He's chewing. He says, oh, that's not
Kind of like your wireless bill.
Whatever.
Mint Mobile is easy,
So,
online, and just 20 bucks a
anyway,
month.
look that
Right now,
up.
buy 1
Chunky
month and get
milk
2 months
commercial.
free
You
at
probably
mintmobile.com.
couldn't hear it very well. I have to figure out how to get the volume to project up there. I think that was just from a couple of years ago. The other thing I don't want to forget to say is the drop date is coming up. So next Friday is that drop date. So what that means is after that date, if you, we call it a withdrawal after that date. It's very difficult to drop the course after the drop date. You have to have a very particular emergency that's happened since that drop date. So just kind of keep that in mind as you're thinking about, goals for the course and where you're at right now. Let me know if you want to run the numbers. I will be away next week and not likely to be able to interact with you very easy. I'm going away on spring break for the first time since 1993. So I'm really excited. So anyway, I'm trying to focus this week, but next week I won't really be around. So if you're thinking about that, let's think about it this week if we can. And if I can be of any help to you, let me know. All right, everybody that took the test, everybody that's going to, I believe, has taken the test from last week. So there were only four questions that less than half of the class missed. So it wasn't, any one or two that kind of tripped everybody up. But this was the most commonly mixed one. And, it's an application question. So you have to really kind of discern, wait, what is this asking really? So genetic deficiency of glucose 6-phosphate dehydrogenase in humans can lead to hemolytic anemia due to inadequate NADPH concentration. Which of the following pathways are going to be affected by this? All right, so that NADPH, which of these pathways involve that? Embden-Meyerhof, that's glycolysis. Okay, so that leaves us with entner-Doudoroff and pentose phosphate that involve that electron carrier NADPH. And then which one do humans do? That's going to be pentose phosphate. Remember, entner-Doudoroff is only some bacteria will do that pathway. Main function of a fermentative pathway. So this was there, like, I think there are 5 different questions that were matching basically which of the pathways does this. So that tripped quite a few of you up, 1 or 2 of those. Main function of a fermentative pathway is to recycle electron carriers. So this pathway can continue to function. So by this pathway, I mean, which one of these? So maybe my pronoun, it wasn't clear exactly what my pronoun was referring to. Correct answer here is we're looking for glycolysis. So Embden-Meyerhof pathway. All right, so those NADHs that come off of glycolysis dumps those electrons off at something like pyruvate, and then it can cycle back. So NAD can keep stripping those electrons off of glycolysis. Okay, so NAD gets reduced to NADH, and that powers some other oxidation reaction. You can't have a reduction without an oxidation, or you can't have the oxidation reaction and glycolysis happening if there's not some reduction somewhere else. Which of the following is correct comparing replication with transcription? More errors are made during replication than during transcription. No, hopefully not. We talked about how the error mechanism, the mechanism of proofreading is much better in replication than what it is in transcription. Transcription involves the use of release factors, while replication does not. Neither one of them do, as far as I know. Both processes are completed with the help of the Rho protein. Sometimes transcription stops with the help of Rho protein, but replication doesn't involve that at all. So correct here is both processes take place in the cytoplasm. Okay, you got to read carefully. I was talking about prokaryotes there. All right, and one more, the mismatch repair that takes place, that remember involves methylation. So if you have, say, the old strand has a guanine base and say the new strand matches up thymine, okay, that's a mismatch. It's supposed to be guanine is matched with cytosine. So these enzymes that move down the strand, how do they know which one to fix? They're going to fix the new one. Okay, so the strand that is not methylated is repaired. Remember, the old one would be highly methylated. Eventually, the new one gets methylated as well, but not until after these repair mechanisms would have a chance to go down. Okay. All right, so be sure to stop by ideally this week while it's still a little fresher in your mind if you want to look over your exam. I did have somebody that looked over his exam and realized that because he skipped a question, it threw off like 10 of his answers after that, and I was able to give him those 10 points back. So sometimes, you know, if you get way worse than what you think you should have, it doesn't hurt to take a look, and we can fix that. So, all right, so continuing to think about ways that we can control microbial growth. So we're thinking to begin with here, we're in chapter 9, and you can be working on chapter 13. We finished up the viruses. Think about physical methods that we can use to control growth, and then we'll get into chemical methods today as well. So we were talking about heat and the application of using moist heat. I tried to look in a little bit about how the pasteurization process is done. Most often it's using steam to bring that temperature up really quickly and then to cool it really quickly again. Maybe like some cold water or cold milk to cool it down again. A third application is using an autoclave. So this is what an autoclave looks like. That's a really kind of dirty looking one, but it's a big kind of double walled, huge, industrial looking device. And it uses steam under pressure in order to sterilize. Okay, so this is a really good method to kill absolutely everything, even endospores. So the parameters that are typically used that are usually good enough to kill anything, we bring the temperature up to 121 degrees Celsius. So that's not the air temperature, that's the water temperature. We always there's a lot of water in the bottom of this in addition to the steam that circulates. How do we bring water up above boiling point? Remember, boiling point is 100 degrees Celsius. After that, the water turns to vapor. But if we increase the pressure, that actually increases the boiling point. So we can superheat the water even hotter than 100 degrees. We bring it up to 121 degrees Celsius. Penetrates really quickly. How much pressure do we add? We add an additional atmospheric pressure. So regular atmospheric pressure is 15 pounds per square inch. We add an additional atmosphere and then leave those conditions for 15 minutes. and that's considered good enough to kill just about anything. Okay. It's pretty flexible in lab. We use this for everything, for our glassware, for media. You know, even things like swabs can be put in there, pipette tips. A lot of different things can go in there. Anything that's heat resistant and that you can get wet. Some things like oils and powders you wouldn't want to put in there. You can't put things like thermometers in an autoclave because they would burst. Sometimes sharp cutting instruments get dulled when you put them in an autoclave, or metal things might corrode, but it's a pretty effective method. Okay, but our autoclave that we have in our prep room, it actually regularly breaks down. So how do you know if it's not working or not? Every time we put something in the autoclave, we add some of this tape that has these heat sensitive lines on it. So the tape looks like this top row here before you put it in the autoclave. And if it comes up to the appropriate temperature, you'll get these dark lines that form on it. So it's not a perfect method. It's quick, you know, easy to do that, but you don't really know how long it's been brought to that temperature. So if you have something that it's really, really important that it's sterile, maybe you've been working with some biohazardous organism or a really important experiment, you may want to absolutely make sure your autoclave is coming up to the right parameters. So you could use a biological indicator also to test if your autoclave is sterilizing. Okay, so how this works, you get a little, you buy a little kit that has a strip of endospores on it. Like it's kind of, you can't really see it right here, but it's like a little paper that's within this plastic vial. And then within the plastic vial here on the outside, there's a glass vial on the inside that has some kind of media in it with nutrients, you know, microbiological media. So you put that whole thing into the autoclave with whatever else you're sterilizing. And then afterwards, you take it out and you twist that plastic part enough so that it breaks that glass tube on the inside and releases that media so it comes in contact with the endospores. And then you incubate it. And there's also a pH indicator in that media so that, you know, after you incubate it for a day, if those endospores have germinated, they'll start to metabolize the media and you get a pH change. Okay, we always have acidic end products when sugars are being utilized. So if you get a yellow color change, then you know those endospores were not killed. Okay, so then you know that the objects are not sterile. Okay, you got to get your repairman in to fix your autoclave. If you got no color change, then you're all good. The endospores are not viable any longer. All right, so we talked about moist heat. Dry heats we don't use as commonly just because it takes longer, but you could use a hot air oven to sterilize something that maybe you couldn't get wet. It does require higher temperatures. So for example, 160 degrees Celsius, that's about 320 degrees Fahrenheit. So, you know, your oven at home would get a lot hotter than that, but I don't know, for some reason, that's the temperature that's often used. You have to leave it for 2 hours. So it takes a little bit longer, but you can sterilize things that way. Again, for things that can't get wet, that might be appropriate. And of course, using heat to burn something, very effective to incinerate trash. Any kind of heat stable instrument, like your loop. Right, you flame your loop in lab and that kills everything. All right, so we talked about using heat quite extensively. What about using cool air, cold air? Cool temperatures are going to be considered bacteriostatic. or bacteriostatic. Okay, that static suffix meaning it's going to stop bacterial growth. Not going to kill things, just going to keep them from growing. So in the lab, when you inoculate your cultures, usually we incubate them for a day or two, and then we put them in the refrigerator so you come back again. Otherwise, your cultures are likely to kind of overgrow. So we like to kind of stop them after a day or so. There are exceptions to refrigeration slowing down growth. Of course, there are organisms that grow best at cold temperatures. We call those psychrophiles. They grow pretty slow anyway, but they can, that's their optimal temperature, like right around refrigeration temperature. And there are some organisms that might be considered facultative psychrophiles. So Listeria monocytogenes is an example of an organism. It's actually a mesophile. It grows best like right around room temperature, but it also grows pretty well at refrigeration temperature. So this organism is known to be a fairly common foodborne pathogen because it grows pretty well in your refrigerator. So you know, after your leftovers or, you know, a certain age, for me, a week is it, and then I throw it out. Maybe that's even letting it go too long. But there's been numerous outbreaks of Listeria. I think the most recent one that really made the news was cantaloupe about 10 years ago. So Listeria is found as just normal microbiota in the colons of livestock, like cows. So it's going to be an agricultural runoff somewhere. I think out in Colorado, they irrigated a field from, you know, water that was contaminated with agricultural runoff, and a bunch of cantaloupes got contaminated. And of course, we usually eat cantaloupes without cooking it. So there was, gosh, like 100 people that died, I think, across the US. And actually, there was a bad Listeria outbreak in the late 90s, I think, at a Bilmar meat processing plant here in Zeeland, Michigan. I think 20 some people died from that. So it's a pretty serious kind of food poisoning. Most people are not going to get terribly ill unless you're kind of immunocompromised already, but it can cause abortion in people that are pregnant. So it's kind of one of those serious ones. Freezing. That's a tangent though, sorry about that. So obviously the colder temperatures we use, the slower and slower organisms are going to grow until they're really just kind of suspended. If we want to save a cell culture and preserve it, oftentimes we'll store cells at minus 80 degrees Celsius. So not all the cells are going to be viable when you thaw it out again, but usually enough of them to start the culture kind of actively growing again. So that's a common way that we use to preserve cells. All right, and some other methods, other physical methods we could use to slow or stop bacterial growth, kill the unwanted bacteria. If we deprive microbes of water, they need water for their metabolism. So that's a good way to control unwanted microbial growth. So desiccation, drying foods out, especially. So here's a picture of some apricots drying in the sun. I don't know if you've ever bought apricots at Meijer. They're really good, but they last when you take them home for like a day or two, and then suddenly they're all soft and rotten. So commonly are going to dry apricots to preserve them. We do that with lots of other foods as well. Let's see. And then lyophilization. Okay, so this is a process that combines freezing. So quick freezing something really fast, like some cells or maybe even some kind of food. And the way we would do that is by dipping it in liquid nitrogen. So you freeze it really fast, and then you put it in a vacuum. So basically you suck the air out of it. So this process, this is a sublimation process where you're taking water as a solid, as ice, and then going straight to a vapor. Okay, so you're skipping that liquid state. And by doing that, you kind of keep the ice crystals from disrupting the structure, and it's going to preserve whatever you're trying to preserve a little bit better. So astronaut ice cream, that's an example of using the lyophilization process to preserve something. Backpackers food, you can buy these pouches of lyophilized food that you just add water to, and they're supposed to be pretty tasty again. Filtration. Kind of crammed a couple things together on this slide. But if you've got something that you need to sterilize and it's sensitive to heat, then this is a really good alternative process to sterilize something. So you use a membrane filter here, you put it right here that has pores small enough to catch anything that you're trying to sterilize. So if you're trying to sterilize something, those pores have to be really small, even to filter out viruses, for example. So say you have, you know, some kind of drug, for example, something that's heat sensitive, you kind of force it through that membrane pore. So you got to use a vacuum to kind of suck it through there. And then once it passes through the filter, it's effectively sterile. So something like a drug or a vaccine, for example, this would be a good method to sterilize it. And if you may remember, let's see, what year was it? 2012. There was a big to-do involving New England compounding pharmacy. They sold this, I think it was a prednisone drug, so a steroid drug that they had not done the proper quality control on. So when you're preparing a drug, you got to take some of it and make sure that it's not contaminated. And they hadn't done that properly. So this batch of prednisone that went all over the US, and some people got it that had back pain, that got it as an injection into their spinal column, developed fungal meningitis. So really serious. There was like 700 people that got sick, 64 people died. It was a whole big scandal. People were jailed. So that one example is one way, again, to sterilize drugs. We could also use filters to sterilize air. So a HEPA filter, again, to sterilize air. Fume hoods in laboratories have air filters in them. And really the mask that we have on, that's just a primitive filter. It's, you know, obviously the cloth ones aren't gonna filter out viruses. They're not that small, but the idea is to catch any large drops that we kind of spray out when we talk, so that those viruses kind of get caught in our mask rather than reach somebody else. So by the way, I was talking to our head of our virus action committee, and he said they're gonna really carefully look at the numbers when we get back from spring break, and there's a possibility that we might get to take our masks off. So that would be really nice. So, you know, there might be a little bump after we get back from spring break, but there is hope that we might actually get to ditch our masks eventually. Cases are way down, so that's good. Okay, osmotic pressure. So this is another way essentially to dehydrate cells, right? You apply some solute that can't pass through that selectively permeable barrier of the cell membrane, something like sugar or salt, create a hypertonic environment. So those cells that would be perhaps, you know, feeding on that food and rotting it are going to dehydrate. So again, hypertonic medium, cells dehydrate. You know, we don't just add sugar to fruit to make it taste good. That's sort of a secondary benefit. The main reason is to try to preserve the food for longer. So jams and jellies, for instance, will last a long time in the refrigerator. Usually fungus are a little bit more resistant to osmotic pressure, so it's usually not going to be bacteria that would eventually make that dam go bad. It's going to be some kind of mold. Okay. All right, and our last kind of physical method here, we want to discuss the use of radiation to control microbial growth. Okay, so, and we talked about this a little bit before because we talked about it as a mutagen. So first, ionizing radiation. Remember, that's bad for cells because it creates ions. Okay. It pulls, meaning it pulls electrons off of other molecules. So exactly what does that do in cells? And we talked about already how it causes a lot of problems, a lot of damage to DNA, even actually breaking both strands of DNA. Oxidizes covalent bonds. All right, there's lots of covalent bonds in things like DNA and proteins. So it's going to mess with the structure of those things. It's going to create hydroxyl radicals. So that's the most toxic of the toxic oxygen forms. Again, pulling electrons off of other molecules. So lots of damage. You know, if you have something that's sensitive to heat, might need to consider some kind of radiation because, again, it doesn't get hot and it has the potential to disinfect or sterilize even really quickly. And depending on kind of what form of it you use, it can penetrate pretty good, at least better than non-ionizing radiation. Okay, so we're talking about these wavelengths here, less than about 10. And shorter and shorter the wavelength of light, the more energy you get. So we're talking about those really short wavelengths. All right, so 3 forms of ionizing radiation, 3 forms that can take, and talk about some of the uses for the different forms. We can use an electron beam. Right, so this would be sort of the shortest of the wavelengths produced by a cathode ray machine. Again, shortest wavelength, it does act really quickly on cells that you're trying to kill, but it doesn't penetrate very well. So you would, that's not intuitive to me. It seems like it's really short wavelength, it would, but apparently not. So anything you want to kind of sterilize just on the surface, you could use this for. So things like spices, some medical supplies are uses there. Probably more commonly used is gamma radiation. Okay, where does gamma radiation come from? It's emitted from radioactive elements like cobalt 40, I think, or is it 60? Cobalt 60. I knew that didn't sound right. So the problem with gamma radiation has a lot of potential to use it. The problem is more the public perception because it comes from radioactive elements. People think that it's going to cause something to be radioactive, which is not the case. That's been shown study after study that that doesn't happen. But this penetrates really well. So it has a few more uses. It takes a little bit longer than electron beams, but again, that penetration is nice. Slow to be accepted again because of this worry that it's bad for us. But probably the most potential use here is in extending the shelf life of our foods. So this is a really striking picture. The 2 groups of strawberries here. This one over here, they hit with some gamma irradiation like a week before that. And then, or whatever, over time, the book doesn't say exactly. Over say a week, the group that hadn't been irradiated, right, that's not fit to eat anymore. So if you it's the microbes that are rotting the food. If you get rid of the microbes, the food's going to last longer. So really has the potential to keep grocery stores from throwing out a ton of food that they can't sell fast enough. So this is kind of slowly becoming more accepted. Any food that has been irradiated is supposed to have this symbol on it. It's called a radura. Just so people know that it's been hit with gamma radiation. The list of foods that is okay to be irradiated with gamma rays now is growing longer and longer. It seems like whenever we have an outbreak of something, like a few years ago, there was spinach, E. coli and spinach. Obviously, that's something that we sell raw, so you can't cook it, or you can, but like to be able to sell raw. So if we could hit it with gamma irradiation, then we could extend the shelf life of that. There are a few foods where it changes the flavor a little bit. Doesn't really change the vitamin content, only a very small amount. So again, medical supplies, Band-Aids, things like that. That's another application, but really potential to use this in foods quite a bit. It doesn't leave any kind of residue either. So, right, you can't, use chemicals because it's something we're going to eat. So that's potential. The last kind of radiation is using X-rays. X-rays obviously penetrate really well, but it takes a really long time for them to actually sterilize something. So that's just not as commonly used in terms of using it for any kind of disinfection. All right, and then of course the other kind of radiation is non-ionizing. Okay, UV light, ultraviolet light, it's an example of non-ionizing radiation. And of course, we talked about this before. It causes a particular kind of mutation, thymine dimers, or sometimes we say pyrimidine dimers. It happens less likely with cytosines though. And somebody asked a really good question. Well, what's the mechanism by which those two thymines now bind to each other? I looked that up, and I can send you a paper if you're curious about that, but it gets way over my head in terms of chemistry. So if you're interested, I can send that to you. But due to the structure of thymine, it's just more likely to happen with the thymines. And of course, that causes a bulge in the DNA, so it's not likely to replicate or transcribe properly. Few drawbacks with using this as an antimicrobial, it doesn't penetrate very well. So ionizing radiation penetrates a lot better than non-ionizing. It doesn't penetrate through solids at all, somewhat through liquids, really good through the air, but not through solids, a little bit through liquids. And of course, like ionizing radiation, it's harmful to humans. So we don't want to reach under a UV lamp that's going to possibly cause damage to our skin. Long exposures could cause skin cancer. But there's still a number of uses. A big one, even though it doesn't penetrate great through liquids, if you can kind of regulate the flow rate, it's really commonly used water treatment plants, sewage treatment plants. So that's what this picture is. You've got the lamps right down in the water. You got to regulate that flow. It's kind of a final step of disinfection and sewage treatment before it's let back out into some body of water. Laboratory equipment, I think over in chemistry, they used to hit the lab goggles with some UV light to disinfect them. And here's another use. I showed this in lab as well, but just as kind of another tool to use in hospital rooms. Of course, we use a lot of chemicals to clean in hospitals, but to try to get into every nook and cranny. Maybe you got somebody with a, tough to kill infectious disease. You wheel the UV lamp in, and then you leave the room and turn it on remotely, right? You especially never want to look into a UV lamp. Leave it there for 15 minutes, turn it off, maybe move it to another part of the room. Again, because it's not going to penetrate through solids, but just another tool to use, another tool in our arsenal. All right, so that's enough of what we would consider kind of our physical methods of control. So chemical control, I think even before the pandemic, as a society, we've been really gone a little bit overboard on chemical control. What's happened again and again, the chemical companies kind of lobby for their use, and then saying, oh, we have to insist that such and such is held to this level of cleanliness. And I think there's a little bit of before the pandemic, there was a little backlash that, hey, maybe we don't need to achieve that level of cleanliness. Maybe some of these chemicals are building up in our bodies, and maybe that's causing some problems. So before the pandemic, there has been kind of a list of chemicals that have been banned from use. Of course, now everybody's kind of crazy about cleaning again, so I don't know where that will go. But we thought before about things that we need to keep in mind when we're choosing which method of antimicrobial control. Good chemicals in particular, we want them to have low side effects. Okay, so in other words, we don't want them to be harmful to us. Ideally. Maybe we have a few in our arsenal that we can use and be really, really careful. But I want them to have low side effects. Ideally, they're going to be soluble in water and useful at high concentrations. Okay, so both of those things. When you're thinking about how expensive something is, if you can dissolve it in water and you can use it as a really dilute solution, then it's going to be a lot more inexpensive. So that's always a plus. And ideally, we'd like to use something that's broad spectrum. Oops. I don't know if I spelled that right. So what I mean by that is ideally, we'd like to maybe control, if we're trying to clean a surface, we don't want to just kill bacteria, we don't want to just kill viruses, we want to kill viruses, we want to kill bacteria, we want to kill any fungi that might be dangerous. So maybe lots of different kinds of organisms. Now when we're taking drugs, that isn't necessarily the same goal. So we'll talk about that when we talk about antibiotics. Sometimes it's much better to target the one thing that's causing disease rather than just trying to wipe everything out. Okay, so ideally it's going to be broad spectrum. So I'm going to talk about a number of different chemicals. I think I've got about 6 that I want to talk about. And I know I'm kind of throwing a lot of information up on the slide, but I want you to pay particular attention to a few things. Okay, I want you to pay particular attention to how these chemicals work. So, for example, are they, interrupting membranes? Are they denaturing proteins? Sometimes we know that, sometimes we're not really sure, but if I talk about it, how it works, the uses of it. So common uses, as well as any drawbacks. Okay, so we're thinking about, well, when would we want to use a certain chemical, and what do we have to be careful of? When is it not going to work? That sort of thing. Okay, and I've arranged them as being kind of low potency to high potency. So low potency meaning it's going to kill those organisms that are easy to kill, like the membrane-bound viruses, the gram-positive bacteria. Those are some of the ones on the lower spectrum. The high potency ones are going to even kill endospores, even be sporicidal. So, okay, and I don't think your book quite organizes it that way, but that's the way I've tried to do it. Okay, so low level. So again, that means it's going to kill those microorganisms that are a little bit easier to kill. Kind of the first thing that you think of when you're trying to clean something is using soap. And soaps and detergents, they're pretty closely related. The difference between a soap and detergent, soaps are a natural product. Okay, so I was kind of looking a little bit up, like how do you make soap? You take sodium hydroxide or lye and mix it with some kind of oil or fats or butter. It's a process called saponification, in case you're interested. But soaps, by definition, they're made from natural products, where detergents are always going to be synthetic. Okay, but they both work kind of a similar way. They are both surfactants, and I edited this slide a little bit, but I didn't really make that very clear. They're both surfactants, both detergents and soaps, and that just means they reduce the surface tension of liquids. So basically, they're decreasing the attraction between molecules. So when we use soap or detergent to remove dirt from a surface, that's also kind of enabling us to wash off the microbes as well. Okay, so when we use soap, we're typically scrubbing. So we're using soap on our hands, we're usually scrubbing. So mostly the action of soap is due to degerming. Okay, remember we defined degerming as kind of a physical scrubbing, for example, is degerming. So it's really kind of less an antiseptic, pure soap, than more of just a way to wash microbes off. Usually say your hands, for example. Now, today, we almost always add some kind of chemical into soaps. It's very difficult to buy a soap that's not labeled as antibacterial because it's got some chemical in it. But really, most of the time, that's not even really necessary. They're going to, again, be antibacterial just because they're washing the microbes away. Detergents, a good example of a detergent, one that's really commonly used because it's very cheap and doesn't leave any residue, is quaternary ammonias. Okay, you probably have heard them just called quats. Really important class of detergents. And the way that they work is by inserting into membranes. So here's a structure of ammonia here. So if you've got a quaternary ammonia, basically all of these hydrogens are going to be removed and substituted out with other side chains here. So here's an example of a quaternary ammonia. The tail here, this is the hydrophobic tail. And hydrophilic head. Okay, so that tail is what inserts into the membranes. So anytime the membrane of an organism is disrupted, it's going to cause all sorts of problems when the membrane's not intact. So especially effective against membrane-bound viruses, bacteria, obviously, especially maybe gram-negatives even a little bit more. Okay. Okay, that's all I wanted to say there. All right, so that's the only low-level antiseptic or disinfectants that I wanted to bring up. There's a couple other in your book. I used to talk about using heavy metals. I used to talk about using phenols, but both of those are not as commonly used just because they're a little bit more harmful to us as humans. So it was getting harder and harder to think of examples of when we use them. So I thought, oh, this is not worth talking about anymore. So we'll move up to the intermediate level and talk about the use of halogens. We're going to focus mostly on chlorine and iodine applications. Some other halogens like bromine. Probably the best application there is using it in swimming pools. It's supposed to be a little bit less irritating to skin than what chlorine is and to the eyes. Fluorine, of course, has been used in water treatments for a long time. It strengthens the tooth enamel and also has some kind of bacterial static properties as well. So you can all thank, hopefully, having less cavities thanks to having a fluoridated water supply. I know my parents didn't grow up with that and they got terrible teeth. How do the halogens work? So if you remember, if you can view the periodic table in your mind, the halogens are over on the right side. They have an almost full electric shell. Okay, so they want to fill that up. Okay, so they are going to grab electrons from other molecules. Okay, so in other words, we could say they're strong oxidizers. Okay, grabbing electrons off of other molecules in order to fill up their outer shell. Okay, so when they grab those electrons, that's primarily going to disrupt those disulfide bonds within proteins. So if you mess up the bonding between proteins, then that impacts their activity. They're not going to function like they're supposed to and can cause some permanent denaturing of proteins. So acting mostly on proteins probably has some other effects as well. We consider them intermediate level antiseptics and disinfectants because their exposure will take several hours to kill endospores. So because they're not killed in sort of a reasonable amount of time, they're not considered sporicidal. Okay, I think you have to kill within 10 minutes to be considered sporicidal. All right, so let's take a look at chlorine and iodine uses. Probably, I know at home, the first thing I think of if I want to make sure something is clean is I bleach it. It's just kind of the easiest thing to use at home. So we'll look at some other kind of forms of chlorine and not just bleach. It's a very strong, usually use this as a disinfectant. So it's not going to hurt you to get some chlorine compound on your hands, but you wouldn't want to do it over and over again over time. It can be irritating over time to our skin. And keep in mind, a lot of disinfectants, you have to clean the area first. So if you got a big mess that you're cleaning up, clean it first and then disinfect it. Because if you just add that disinfectant and you clean at the same time, it doesn't penetrate very, very well through organic loads. And something to remember for chlorine compounds, they're very sensitive to light and pH. So if you ever wondered why we buy chlorine in these opaque bottles, it's because they're going to start to go off when they are exposed to light. And they will anyway. Like a bottle of bleach like that only stays active for maybe like 6 to 9 months before it starts to go off. So if you're trying to, whiten your whites and your bleach isn't working, it's because you probably had it for too long. So I always buy bleach in small containers, try to use it up within a year or so. If it's really old, it just doesn't work very well. Okay. As well as pH. So we mentioned this before. If you've ever, been in charge of taking care of a swimming pool, if the pH gets too high, that chlorine dissociates into a form that doesn't work, and you can just, you know, dump more and more and more chlorine in it, and it's not doing anything because your pH is off. Okay, so we see some different types of chlorine. We can use it in the form of a chloramine. So that means here's an ammonia molecule. That just means that either 1, 2, or 3 of these hydrogens is replaced with a chlorine atom. Why do we do that? It makes the chlorine a little bit longer acting. It's just kind of stays around for a little bit longer, works for a little bit longer. So don't confuse that. You never want to mix bleach and ammonia as cleaners. It's actually kind of difficult to even find ammonia at the grocery store. But if you mix bleach and ammonia, it makes a vapor that's toxic. It can actually kill you if you do that. So you don't want to mix those two, but if they're kind of manufactured or synthesized together, then, you know, apparently it works fine. And it's the vapor, again, that's toxic, not the liquid. So we've seen chlorine in all these different types. Really, there's, in terms of when we use one type and when we use another, about all of them are used for water treatment. So that's a really common thing. In terms of, you know, sewage treatment and drinking water treatment, that's really the most common use. And when you would use one and when you use another, it's really not clear to me. But we could also use it in the form of elemental chlorine. Hypochlorites, that's another name for bleach. Okay, and again, bleach, great disinfectant. It's one of the disinfectants that can be used without very much contact time. So pretty much kills pretty close to being on contact. It's probably better to give it a little bit of a soak before you wipe it off, but really, really fast killing power. Again, it's very cheap. If you want to disinfect maybe a big space, then you could use chlorine dioxide gas. Okay, so lots of advantages there. And the other halogen that we'll talk about here is the use of iodine. A lot of antiseptic uses. So remember, disinfectants on non-living surfaces, antiseptic, designed to be used on living surfaces. And again, we can find iodine in several different forms. We'll talk about that. Commonly as some kind of wound treatment, or maybe before surgery, before some kind of needle poke or incision. You can buy iodine tablets to treat your water. So maybe you want to go camping in the backcountry, you don't want to carry your water with you, so you take iodine tablets. The problem with that is that at the concentration you use to make the water safe to drink, it doesn't reliably kill cryptosporidium. So remember, cryptosporidium, that's that protozoa that causes really nasty diarrhea. It's pretty resistant to both chlorine and iodine. We could use enough iodine to kill it with tablets, but then it wouldn't be good to safe for us to drink. So that's the one drawback to using it to treat water. It does work a little bit better in any kind of organic load, so it has that advantage. But of course, if we're going to use some kind of iodine on a surface, it's staining. All right, so that really limits. You don't want to wipe iodine on your kitchen counters every day, right? Because then they look nasty. With any chemical, you can have some minor allergies, but maybe there's a little bit more with iodine. Oftentimes, we'll use iodine dissolved in alcohol. So we call that a tincture of iodine. Makes it, I think, a little bit more penetrating. But probably most often we'll use iodine in the form of an iodophore. And that just means the iodine has been complexed with some kind of carrier molecule, usually a protein, that will make it longer acting, as well as less irritating to skin. Okay, so the form that's usually used, you may have heard of something called povidone or povidone iodine. The trade name there is betadine. Okay, so betadine is just elemental iodine that's been complexed with a carrier protein that makes it just a little less irritating to the skin, a little bit more longer acting. So in this picture in your book, a patient's hand is being basically washed in betadine before a hand surgery. All right, so it keeps up that antimicrobial action over a little longer period of time. So lots of medical applications, you know, before a blood draw, something like that. Another application in the dairy industry. I visited an aunt and uncle a number of years ago and saw them using this. It sounds a little crude, but it's called Teat Depth. All right, so they go down, and this picture is like the cleanest cow I have ever seen in my life. But they go down, and I don't know if they hose down the cow a little bit first, but then they want to clean the nipples before they milk the cow. They use this betadine solution that they call Teat Depth, and they go down and, you know, do maybe like 6 or 7 cows, and then they'll go back to the beginning and wipe it off. So they leave a little exposure time. They don't want it to obviously drip into the milk. Once they take it, they wipe it off again. But again, just one more way that we can reduce the number of microbes in raw milk, so that, you know, when we pasteurize, we reduce those numbers even more. Okay, so one more intermediate level antimicrobial. Lots of applications here. Use of alcohol. So I'm primarily talking about isopropanol, or also called isopropyl alcohol, also called rubbing alcohol, and ethanol. Okay, so I was looking up, I have isopropyl alcohol at home, and the main thing I use it for at home is whenever I take somebody's temperature, is to clean off the thermometer again. But it has lots of, you can use that to clean anything, mostly again as a disinfectant, surfaces. I was also reading that because rubbing alcohol has such a low freezing temperature, that you can mix it with water, and if your windshield has got a layer of ice on it, you can pour it on your windshield and it will melt your ice real easy. So this ice storm, you know, that's an idea rather than kind of chipping it away forever. There are antiseptic uses. It depends on the concentration that we use. So for that, of course, everybody is familiar with this by now, but you know, hand sanitizer, right? That the active ingredient in there is alcohol, and everyone that I've ever seen, somewhere between 60 and 70% alcohol in those hand sanitizers. So that's how we can use alcohol as a antiseptic. Again, if you use it more concentrated, it could be considered a disinfectant. It's going to work primarily on membranes, but also it's going to denature proteins as well. And we usually use it, like, this is the one exception. So I said before, the more concentrated a chemical is, usually the better it works. This is the one exception. You don't want to use 100% ethanol, because apparently a little water is needed for that surfactant action. Okay, so a little bit of water is necessary, so it works as a surfactant. Meaning a surfactant means it dissolves kind of the bonds between things, reduces that surface tension. And also 100% alcohol evaporates really quickly. So as soon as you add it to a surface, it's gone right away again. So It reduces a little bit of that contact time that you need. It's, you know, it's cheap. It doesn't irritate our skin. The only drawback is that if you're exposed to huge amounts of it, it's an inhalation hazard. And that can be bad, but you know, the amounts that we're using in hand sanitizer, it may smell kind of nasty, but it's not enough to be a problem. All right, so I have to ask the question, you know, so, okay, we're talking about hand sanitizer. What's better, using hand sanitizer or washing your hands? Okay, so let's define how to use them correctly first. If you use a hand sanitizer, you have to use enough to wet your hands for 15 seconds. Okay, so we need about 15 seconds of exposure there. And if you're going to correctly wash your hands, you're supposed to do that for 20 seconds. I admit, I don't usually do it that long. And of course, you get some scrubbing action there. Okay, so talk to your neighbor. This would be a good clicker question. Talk to your neighbor. Which do you think is better? Which is more effective? Okay, so talk to somebody near you and see what you think. Do you have dairy cattle? No. Okay. Oh, okay. All right, so I think I mentioned this maybe before. So I heard a lot of people kind of, I heard the right answer from a lot of people. Let me give you a little more information. So one thing about hand sanitizer, I think I mentioned this before, it's not very effective when your hands are dirty. Okay, so that's a drawback. And another drawback is that spores and naked viruses are not reliably killed. Okay, so knowing that, actually it's better to wash your hands if you have access to soap and water. If you don't have access to soap and water, then by all means, hand sanitizer is going to do a lot of good. It's going to easily kill COVID. It's going to easily kill influenza. Something like norovirus, which is a naked virus, not as reliable. So, and of course, you know, you're working outside or whatever. Again, if all you got is hand sanitizer, by all means use it, but it's not going to give you quite as good killing as that washing away of all the germs. Okay, and neither one are going to sterilize your hands. You have lots of normal microbiota on your hands that, you know, you might wash some of it off, but there's still going to be more there. Okay. All right, to finish up, I've got a couple of high-level disinfectants that I want to talk about. The first is hydrogen peroxide. Okay, H2O2. And this has been called the closest thing that we have to an ideal antimicrobial. Because it's inexpensive, it doesn't leave residue. You know, you can use it on pretty much any surface. The reason that it kills pathogens is because it forms hydroxyl radicals. All right, so in other words, it's an oxidizing agent, like the halogens. Okay, it's grabbing electrons off from other molecules, and it just does a lot of damage. It breaks down membranes, it denatures proteins, it does quite a bit. Probably the only disadvantage here is that it's photosensitive, just like bleach. So you're always going to buy this in dark bottles. It will start to, again, it just breaks down and it dissociates into something that's not effective when it's in sunlight for very even short periods of time. And we can use it depending on the concentration as either a disinfectant or an antiseptic. Usually you buy it 3%. I think it's hard to see that 3% is usually the way you buy it, like in the pharmacy. And when I was growing up, that's what you commonly use. You fell down, you skinned your knee, you put some hydrogen peroxide to clean that out. We've kind of moved away from using it as something to clean a wound. More often we'd use something like triple antibiotic ointment now. The reason we've moved away from it is it's fine to do that, the first time, you break the skin, clean that out really good. But then to clean it going forward, you don't want to keep adding it day after day after day, because it's harmful to our cells as well, and it will slow down the healing process. So you're more likely to get a scar if you keep cleaning it over and over again with hydrogen peroxide. So we more often would use other things, but it's fine to use it at least once on a wound. If we use hydrogen peroxide more like 30–35%, then it's considered sporicidal. Right? We might use that to sterilize delicate instruments, medical instruments that you can't autoclave. Right? And one other high level disinfectant, and I don't think I put this one on your learning objectives. I decided I'd cut it before and I decided to add it back in. So it might not be there, but the aldehydes. These maybe have a little bit less use because they're just really strong. But again, these are also considered sporicidal. So especially things like medical instruments that you can't put in an autoclave, things like an endoscope, for example, soak it in something like glutaraldehyde for 10 minutes or so, then that's going to be pretty effective. So how does it work? Cross-links proteins and DNA, very broad spectrum. It does retain its potency pretty well. A real drawback is, especially formaldehyde, it's carcinogenic. All right, so we really have to be careful about its exposure to us. Glutaraldehyde is a little safer for us to use, but it's a little bit more expensive. Again, often use that to treat medical instruments, for example. Formaldehyde is actually a gas. It can be used in its liquid form to preserve specimens, for example. And actually, we often use formalin, that's formaldehyde in its liquid form, as a way to deactivate viruses or kill bacteria before we use them in a vaccine. Okay, so it is found in really, really tiny, minute amounts in some vaccines. Again, used as kind of a killing agent to deactivate that virus before we use it in a vaccine. Okay, questions about any of those? I know sometimes I get through these lists of things and it gets a little bit dry, but let's see. I think that's all we have there. So let me just start our next unit here, kind of introduce it. And our next unit is antibiotics. So we'll just, again, scratch the surface today, and that will probably go into Wednesday and then maybe a little bit into when we come back. But I always like to start my discussion of antibiotics with a little public health lesson and think about what kind of things kill us today. And I didn't post these first few slides because I don't want it to be something that you memorize. What kills us today versus what killed us 100 years ago or 100 plus years ago. So if you look at 1900, the top 10 causes of death, a lot of them were infectious disease. So number one cause of death, pneumonia, influenza, TB, gastrointestinal infections. So that's things like typhoid, something that we see almost none of today because we treat our water. We still had heart disease, strokes, nephropathies sometimes can be infectious. Diphtheria, that's an infection that's kids are a lot more susceptible. It forms this membrane in their throat that basically they can't eventually breathe through. So back in 1900, about 1 out of 5 kids died. No, yeah, 1 out of 5 kids died before their 5th birthday. Okay, so that's a pretty astonishing statistic. We had a lot, really high mortality rate for kids. Sorry, I knew that didn't sound right. 1 out of 3 died before their 5th birthday. So more recently, and I wish I had statistics that were a little bit even better, more recent, but as of 2010, at least in the US, the only infectious disease we had was number 9 here, pneumonia or influenza. Okay, so other things like COPD and stroke, cancer, heart disease, much more common. So as of 2010, life expectancy for white males was 74 years. And if you compare that to 1900, life expectancy 47 years. All right, so we made huge gains. That statistic can be confusing. That doesn't mean that people lived till they were 47 and died, but because you had so many kids dying young, that balanced out the people that lived till they were 80, and you had, people dying more like, an average that averaged out to 47. So, we've made a lot of gains. And I think more recently, at least as of 2019, the average life expectancy was more like 78. Of course, it's different depending if you're male or female. These are the best numbers that I could see recently, and these numbers are a year old, unfortunately. But of course, the pandemic has influenced quite a bit our death rate, and as of last January, that COVID was the -- 1 cause of death. So that's kind of sobering. It'll be interesting to see once everything's all said and done, what they have to say about that. But thinking about why have we, at least pre-pandemic, gotten, why are we living longer? Well, again, I wish I had more up-to-date things, but if you look at some of the things that have been put in place in terms of public health. So about 1900, most states had health departments. So the health department basically educates people on how to live healthier life, right? So we started to recognize that that was important. We started to treat our water supply. Of course, we had a huge outlier there with the worldwide flu pandemic. But because our hygiene was getting better and better, our death rate was going down. So again, treating our water was a big thing there. The last human-to-human transmission of plague. Mostly plague, we can get rid of that just by having good hygiene. So it's carried by fleas that are most often carried by rats. And then in the 40s, our first use of penicillin. We had some really effective vaccines coming out. So those 3 things, better hygiene. So we just got done talking about ways to cut down on microbial growth. We're going to talk about antibiotics next. And then after break, we'll get into immunology, which will lead to our discussion of vaccines. So those are the big 3 things. So, okay, that's all the time we have. I'll see you Wednesday. We'll continue with antibiotics.